Okay, so yeah, so let's start from our first topic. I think we have already covered this BI sequencing in the class. So in this lab, I'll basically go over all the things you would need to you would need to finish the homework. The software that we are focused on, there are multiple softwares out there. So there we pick a specific one. It's called Bismarck, and it requires some specific environment, which we also created on in the shared folder, so that you can directly start from there. We think we lock the version, we lock the file writing permission on the shared folder in the shared folder. But please try not to make any change so that the other students can also use the same file. Okay. So the main schedule is basically like this. For the following 2 weeks, I'll be taking charge of the labs. As always, the Tuesday ones will be interactive lab tutorials, and the Thursdays will be the Q&A sessions. So I try to separate the materials into 2 parts so that we can focus on each of them separately. If you have any questions, working on the homework, which by the way, I recommend you to start earlier. Please just as always, feel free to use the Slack channel and trying to post it there, and with your screenshot, with your scripts and everything, so that we can take a look at before the lab, before the Q&A session, and also other students from the class can also help, and they can also some of the questions might be you never know, some of the questions may be shared between different students, and you definitely usually not alone. So if you post it there, it will be helpful for yourself and also helpful for the other students. Okay. So the first topic we have here is the BI sequencing. Just a very brief recap of the technology. Is basically using chemical a chemical tool that is you can protect this methylation. You can use this methylation group to protect the C from your genome, and you do this bisulfite procedure, let's say, and then you do all this transcription and duplication, and in the end, when you do this sequencing, you basically change the original genome. You change all the CG pairs into TA pairs, but only those that are protected by the methylation will not be changed, so that will remain the same. So by comparing the outcome, the result genome to the original one, you will be able to see where are the methylation events happening. Okay, so on the right side is a more detailed diagram about if you start from different condition like methylated versus unmethylated one, and then you use the same molecule, is using the same reagent to process. So in the end, if you have this methylation, if you can see my mouse here, if you have this methylation, and that will not be changed, after you do this bisulfite conversion. But if you do not have this methylation group protecting, that will be changed into a TA pair. Okay. So that's kind of the idea behind it. But in practice, now that we know this technology, that could work theoretically, but in practice, what are we really going to do? What would be the difference, from doing, for example, genome alignment and mapping? What would be the difference doing this from what we did before? So we can pause here for a second and think about what would be the additional things we need to do. Apparently, here, since we are going to change the genome, we are essentially also changing the reference, the background behind it. So whenever you do this conversion, you will not be able to realign your sequence, your reads back to the standard genome, because imagine in the standard genome, you wouldn't have such a fragment. You wouldn't have this sequence. So the first thing that if you want to use this BI sequencing, and also for your homework, the first thing you need to do is to make a conversion of your background genome, of your reference genome. So starting from there, Then you would do the regular mapping, regular alignment as before. We can use whatever tools that is available. So here we are going to use this Bismarck software, which is a kind of a bundled up a function where you can do multiple processing steps in one line of command. Okay, so after you do this alignment, you need to do duplicates removal, extract methylation, and everything. And also this Bismarck also helps you to generate kind of the output report, the log file, which we request you to append into your submission of the homework, so that you don't need to work on that on yourself by yourself. Okay. So the way I want to do this lab is to lead you guys step by step. Let's imagine we are working on this homework right now. Let's imagine we have a BS sequencing file now sitting on our flash drive. What should we do with it? So what would be the first step? What would be the second? And then I really believe my philosophy is really is learning by practicing. So hopefully through the process you'd also have a better understanding of the theory and of the theoretical algorithm behind it. And also please feel free to interrupt anytime if there's anything too technical or is anything not that clear. Yeah, so first thing we need, as we need before, we need some alignment tool. So we use this bowtie indexing file. I think we used them before. So we are going to use that again. So since we are using this Canon, well, apparently you can use your local machine, but we recommend you to use the Canon server. Then you would have to go through their module manager system. So you would need to load this bowtie, load this package called HESAT and this SIME2, which is a kind of a basic box, say, for genome alignment. For this software of interest, this Bismarck, we leave it to you guys so that you can download and install it by yourself, because this one is not actually a package that need to be pre-installed into your environment. It's more like a folder of commands that you can directly use directly. So as we put in the homework, we ask you to go to this Bismarck document to see how to use them and also how to install them directly from scratch. Just one thing to note here is that this Bismarck software is written in Perl. So try to call it cleverly, because usually the functions we call from the bash, for example, Python here, they are all pre-compiled into a binary file, so they can call this Python as a command directly. But imagine let's say if we go to this Bismarck, so you would have a list of commands that you would need most of them. For example, this one, after you finish doing all this Bismarck thing, you want to generate a report, you want to generate a summary, and you want to call this function. But here we will not be able to say Bismarck, because that's not a command. So in order to compile this thing, you can say there are two ways, basically. There are multiple ways. So the two ways I would recommend is one, to use Perl here. It's just like what we do with R or with Python. We put the command first, we put the language first, and put the script first. Okay, and then that would give you the command and all the help you would need. If you look into this this file, it's actually a script file. So it is actually specified here that to use Perl to call this script. Okay, another way to do it is to specify the full absolute directory, the absolute location of the file. And by doing this, you will also be able to call this function, to call this command directly, because the bash would help you to do this compilation. I see some conversation in the chat. Let me see. Yeah, by default, if we do not specify, try to install everything on your server, in your Canon home folder, or in your Canon wherever you want to put your working directory to be. You can, as I said, you can definitely do everything locally, but we don't recommend that because that would be computationally very heavy, and also would take a lot of disk space. And also, we already set up this environment on the server, so that we're trying to make a uniform environment so that we can all start from the same page. Otherwise, you might have different versions, and etc. Okay. Yeah. So that's how you call the Bismarck commands. So with all this, I think you'll be able to follow the document that is written here. You know, they have different multiple different tutorials, like step by step. So you just follow their steps. For this genome preparation one, so if you go back to our homework here, we also put in step 1, step 2 things so that you can best stick onto the kind of the plan we expect you to follow. So the first thing you need to do is to download and trying to set up your working environment. And then the second is to prepare for the genome. So here, the genome preparation is the conversion, just we just talked about a few minutes ago, is we put it here as a graduate student only, but for undergrad student, we also highly recommend you to trying to test it out, to check it out, to see, what is really going on, what is this Bismarck is really working on. So, just to give you guys a hint, this function, this command, the entire software is pretty well developed and pretty well maintained. So you can directly use a single command and that give you the preparation. Okay. For undergrads, or if you do not want to worry about this, we also provide a version here. That will be a converted genome version, and you can continue to work on the following questions, just based on this converted one. But again, I would highly recommend you guys to check it out. Okay. Yeah. So actually, by the way, let me show you what is the folder looks like right now, so that you can have a better understanding too. It's taking longer than it usually does. Yeah, so this is what the folder looks like right now. So, instead of having all this Markdown file and HTML, we also, in addition, we also have this 3 different data folders and 4 different environments. So try not to worry about the other environments and only focus on this conda environment 37. Because later on, when we do this Python code, especially for the part 2, we would need a lot of dependencies from Python. So you will need this environment. You can activate this conda environment as what we did before. Okay. The thing that is more you care more about is the data folder. So for different questions, part 1, part 2, part 3, we will have different input data. And for example, we just talk about this Bismarck question. So you would see here, for example, this expression location, which is a profile, a background profile of the general gene expression levels for this, I think it's prostate cancer cell line. Yeah, here. So when you see something like, copy the file, you should be able to directly find the files, including all these byte files, all the background gene expression. And here, this HG38 is the converted version of your genome for this test. And if you want to do this conversion, by your own, we would need you to download this the raw genome from the homework one, instead of using the one we provided in P1 data. Okay. Yeah, so after you finish running all this Bismarck, commands, including, preparing for the genome, running the main function, and then just, deduplicate the Bismarck and everything, and, extract the methylation. You just do that. You just follow the, tutorial step by step. And then in the end, you will be able to, yeah, you just do this step by step. I try to put a kind of template of commands here so that you can directly refer to. You'll be able to call this Bismarck report function, and this report will gives you something like this. This is, if you see something like this, you succeed. Okay, so this is something we would like to see from your submission of the homework, and then directly from this table, you will be able to directly read, for this specific experiment, what are the percentage of CPGs, what are the percentage of the methylated CPGs, for example. And do we see any clear pattern between CPG and CG? So that you can directly answer the question. Okay. Yeah, so that's all we have for part 1.1. Any questions so far? Okay. If not, let me just pause this recording so that it can be separated into sections. Okay, so now for the second part of the first part, okay, part 1.2, we need to calculate. We provide, what we provide is a background of your gene expression for one specific sample, and we also provide you, where are the methylations located, which we generated actually from the first question, right? So after the first question, we will be able to see all these methylation events through the entire chromosome, through the entire genome. And now we also have the background gene expression level. Now we want to see, what we want to check is actually, do we have a lower gene expression if we have a higher, more frequent methylation events? Because that's kind of the hypothesis here, right? If you have methylation, through the chromatin regulation mechanism, you would have, in general, lower gene expression level. We also put here some, kind of positive controls, like wherever you have this histone modifier, those proteins, those enzymes, you would also be able to see some upregulated gene expression. So we ask here that, just trying to state the problem, computationally, what we are doing here is, like, we have a background gene expression and we have some location files. Now we want to look at the intersections. We want to see the overlaps of the methylation events and the gene expression, and the genes, and we want to get the expression level of those genes. Okay, so the tool we are going to use is the BAD tools. I'm actually not sure whether you have been working on this before in the previous homeworks, but it's fairly straightforward. You just load this tool, which is pre-installed already from Canon, and you call saying, I want to calculate the intersection between the A file and the B file. Okay, and the A file, you just put here the background gene expression data, and the B file would be the BAD file, which is the file that record all these locations of your events. Okay. And then we would ask you to write all those files into a new file. Basically, we want you to create a file and save all this output, and then later on you would need those output files directly. So here, just a very brief recap of the batch scripts. So using this vertical line symbol, is to basically, is the batch language trying to append additional commands after you finish the first one. For example, let me see if I have some example here. Actually, we don't. Now imagine, say, I want to take a head of this file. Okay, imagine this is something like we want to do. Okay, so now basically what this cat or what this head is doing is just to pick the first of few lines, and the output of this function is a text stream. You have all this text. So next thing you can do is, now I can search for a gene of interest. For example, I'm specifically interested in this gene, and then I can just grab it. So what this is doing is, you're taking the output of the original function, the previous function, and you feed into the second function, the second program. And the second program will take that as the input and gives you whatever you would need downstream. Okay, so other things can also work. It's like, imagine now I can cat the entire file and then do the head. which is exactly the same as what we did here. So just taking the head directly. Or I want to catch all the expression, all the files. I want to catch all the files. And I want to first search for, all the chromosome 1 genes. Okay, and that will gives you a lot, right? Okay, it also gives you 11, let's say. This would gives you, okay, let's try chromosome 3. Okay, so that would gives you only chromosome 3. Okay, so now you're interested in the first 100 genes on chromosome 1 on chromosome 3 that we have from this expression. Then you put another vertical line. It's just like the the commands we use in R. You can just append whatever you need. So imagine here, now I want to see the head. That will gives you the first 10 chromosome 3 genes that we found from the file. Okay. The reason we are reviewing this is because we will need this for your homework. Because imagine this bad tool, similar to catch, would gives you a output stream. And there are two ways you can do it. One way is you just print it out on your screen as what this cat is doing, right? And then you can copy paste manually from your command line window to a text file. But apparently that's not very efficient, because especially now the files are generally very large. So the way we do that is to say, we can take the head. Imagine this is what the output of the bad tools is. Okay, we do some bad tools, that intersect a file and -b file, and that would give you the string, the string output, that stream. And then now I want to say, calculate how many lines do we have. So that will be a word count, -l. So that will give you how many lines do we have from this. So, in a biological, from a biological perspective, that's answering the question that how many of the genes that we identified as overlapped with the methylation events. Okay. So that would gives you an idea of whether your analysis is making sense or not. Imagine that if that gives you a 0, that means you don't have any output. So the bad tool is not working properly. If that gives you a, I don't know, millions or billions, then that also not reasonable, because we don't have that many genes. Okay, so this is like a sanity check. Another thing to keep in mind is that the bad tools would not automatically remove all these duplicates, because we would have many more methylation events, but we only have 20,000 genes in our genome. So there would be a lot of duplicates, let's say. And then what we need is actually only the unique samples from this library. So we can put, similarly as what we put before, like the head, we can put a unique here. This unique here would just delete all these duplicated lines from your file. And then you can count it again, saying, this asking how many unique genes we identified that has an overlapping region with the methylation event. And eventually, you want to save all these things into a file. Then you put this larger symbol, this greater symbol. That means I want to put whatever I got before this symbol into the file I specified after this symbol. So this so if you run this, you would get the intersection first, remove the unique, remove the duplicate, and save it into a TSP file. And you would need this for your later plotting. Okay. I see another question. Why are we doing the conversion again for part 1? Yeah, let me come back to that. Okay, after we finish this part 2, because it's slightly recorded. It's being recorded, so it's kind of slightly off. Okay, but I will come back to that. Don't worry. Okay, so that's the bad tools. The main function of the bad tools. Now after you do this intersection, you would have for each event list and each gene expression background file, you would have a result file generated with all this overlapping. And then what we are basically asking is, we have designed 4 groups of those events. Methylation is one type of event. We also look at, for example, this H3K27, gene enzyme events. Okay, this modification, this histone modification events, and also the other types of methylation modification. So we want you to compare between this group, the methylation group, and the background, which is the general random background. Okay, so we want you to compare between these four groups to see if you see any consistent gene expression level change or not. Okay, so that's what we basically ask you to work on in part 1.2, which is actually it's a very routine kind of procedure if you want to work with this kind of data in the future. I think this now this time the questions we put in the homework is very hands-on and probably can also be very directly usable when you work on your own projects. Okay, any questions specifically for this sub question? Okay, if not, let me stop sharing this and let me stop recording this, and then we can come back to the question from Yao. Here we go. Okay, so this is actually the final topic we are going to cover today, which is the Hi-C. Again, I don't know whether we have already covered this in your lecture or not, but I think I noticed there are recorded version of the lectures on this topic. So feel free to check it out. So here I just post a very brief history of this technology, starting from now it's like 20 years ago, the first version of trying to get this chromosome conformation, and that at that time it's called CCC, so 3C, where you're trying to pick one gene versus another gene. You're trying to look at the pair of the genes or the regions, and to trying to see if you have any interactions between the regions. Yeah, by the way, Hi-C is just this technique, and trying to imagine in your nucleus all these chromosomes are complete a mixture. And the hypothesis here is that some of the genes can interact with each other, or some of the genetic elements can interact with each other, even across chromosomes, by this spatially getting closer to each other and trying to interact with each other. And then the basic idea behind all these techniques in Hi-C, 3C, 4C is that we artificially introduce some of the cross links, when all the chromosomes are in a mixture state. And then the chromosomes that are closer to each other would have more cross-link events happening. And then now you lysate it again, you digest it into fragments and you do sequencing. And then the difference between the 3C, 4C, 5C is different ways to identify what the sequence are. The sequence can be array-based or can be qPCR-based, which is 3C actually, or nowadays a more modern way, more than 10 years ago or 10 years old already, more modern ways to do this next generation sequencing, so that you can directly take the entire sequence of the cross-linked pair of genes. And then you can imagine the reads now we have is would include two parts, one from gene A and one from gene B, and they are cross-linked with each other. So now what should we do with this kind of data if we want to reconstruct their original structure? So imagine now our input are gene A and gene B fused reads, and two, we know where this gene A is originally from, and we know where this gene B is originally from, because we have the reference genome. We can map it back to the reference genome. These are the two information we have, and the output, the result we want to have. The question we are asking is that originally, spatially speaking, which of the genes are closer to each other? And the idea behind the hypothesis behind this is that if two genes are more frequently closer to each other in the spatial in the space, then these pairs of genes are more likely to have regulatory effect between each other. Okay, which is still a you know an open-ended question, but that's the idea behind it. Okay. Sorry. Yeah. So for this one, we would need a package called run HiC. Namely, it's a package for HiC. So that would requires multiple different dependencies as the supportive package. So I would say I would not recommend you to try this on your local machine. So try to do this in on Canon. Use this environment that we generated already for you. So like we said here, sorry. So in the homework folder, you have multiple different environments. So for this question specifically, we want you to focus on this Python 3.7 version, because that's what the run HiC requires. So you do activate this in the environment like what we did before. Activate, and you copy the path here. Okay. And you copy this, and you also load. Before you are able to work with Conda, you also need to load at Anaconda at Python before you're doing this. So always remember to load this before you're doing that. I'm doing here is because I've already loaded this before. But if you do not, if it tells you basically I don't know what Conda is. Okay. And also similarly, we also need some genomic bioinformatics tools to support. Some of them include SAM tools, Minimap, and BWA. So the BW here is a kind of an alternative version of the Bowtie. It's an index generation software. So we also put in the homework asking, what is the the difference between BW and Bowtie. So I wouldn't cover too much here. But just very briefly, that would really, each of them would have different scenarios that each of them would be better fitted to. I think one major factor people concluded, empirically, from experience, is the length of the race. Okay, I'll just put a hint here. We also put a few additional reading materials in the question here. Actually, I think I put it here. Yeah, so trying to read through their conversation and to get a really understanding of the history, and also get an understanding of what the question is really about. Okay, so now after you you load this environment, after you understand what is the difference between BWA and Bowtie, another optional thing we would again recommend you to try is trying to build this index, use BWA, which probably you have already done before, because in homework 3 you are using this BWA, whereas in homework 1 you are using this Bowtie. So you actually have already finished both. But here, I just want to give just want you to clarify what the difference between them. Okay. After we finish all this, again, this is optional. You don't have to do this. We have already generated the index for you. You can load it directly from a similar folder as as a HG38, but not this time under the P2 data. Okay. So this folder will look something like this. So if we go to that folder, this is the the kind of the prerequisite for run HiC. Another there are quite a few artifacts of this run HiC, because this run HiC is not a it's being actively maintained, but it's developed by a single contributor, or quite a few contributor. So try to be be cautious about their document, and when you're trying to use their comments and tutorials. And for example, here, the reason I'm saying this here is because imagine actually we allowed HG38, the genome, to be in FA or to be in FASTA file. They are actually essentially the same, but this run HiC requires it to be FA, and FA has to be. There's no way to specify this actually. So this is just one type of artifact you would have, you might encounter when you do this homework. And that is also in part the reason we recommend you to start working on this homework earlier, because now we are looking at a lot of third-party packages. So once you get all this, index file and all this reference data, prepared, some of them are from the Canvas, some of them are from, the shared folder. After you set up your working directory, remember to put everything in the same folder, in the same directory, which is a second artifact. So it requires you to put everything all together. There are other ways to do it, but we don't recommend. So try to put everything in one folder, so that make your life easier. And then if you have your working directory looking something like this, you are good to proceed. If you don't, go back to the previous steps to see wherever that is off. Okay. After you prepared for this, you would be directly able to call this run HiC. Now, let's go to the document so that you know what I'm talking about. So this is the run HiC document. So it is also asking you to prepare for the data, which we have done for you to prepare for the genome index and the mapping. Now we are doing this mapping thing, using this run HiC mapping. Remember to call this run HiC only after you activate your Conda environment. Otherwise, you wouldn't be able to, like your system wouldn't be able to recognize this command. And now it will require you to generate some of initial files for like the metadata configuration. And you can use either of this or the other, the following ones to do this mapping. Okay. After you do this mapping, that will generate a folder called pairs, and you would need these pairs to do filtering. And after this filtering, it would generate some filtered folder, and you will need this filtered folder for binning. Okay. Fortunately, the run HiC authors, they have developed this bundle up question, like pile up question function, so that you can directly call the function you call it once and would generate everything for you. But if you run into any issues or any bugs, you might want to go back to the previous steps to really debug step by step to see where it's really going wrong. But hopefully, you can directly call it and successfully, and that will generate to you some statistical table summaries and also some plots, and you just need to put them into our homework so that we know you succeeded in working with run HiC. And then based on those plots and tables, you can answer the following questions, those biological questions. Yeah, again, here, you can do run HiC step by step, or you can do this using this bundle function directly. Again, remember to set up your environment, have at least these 4 information. Okay, data set, the chromosome sizes, the HiC subreads, which is your input, and your reference genome, all in the same folder. Okay. Yeah, and after you run all these bundle functions, you can call this. Well, you have to call this quality function to generate the analysis of quality from your mapping and filtering. After doing this mapping, filtering, duplicates removal, we will be able to look at the actual code, that's the actual data we get from the experiments. And now this quality is giving you the first round of analysis, saying what is the quality of your HiC result, what is the percentage of this and that. And if you look into that, you're looking into them, looking at all these figures, would gives you a better understanding of what the HiC is really working on and what the HiC result would look like. Okay, that's all I have for this part 2.1, and that's actually the last topic for today's lab. Are there any questions? Very nice. I like this silence. The silence means we basically go over the things carefully, and we hopefully you can follow up follow the instructions and not follow the templates of the script from the slides when you are working on your homework. Okay, so in the very end, just a reminder, on this Thursday, coming Thursday, 8 p.m. Eastern time, we'll be having this Q&A session. And this Q&A session would be on the topic that is covered today. So for the other part of HiC questions and also the hidden Markov model questions, I would recommend you to save it until later, because I believe on Thursday we will be focused on the first two topics. And also on next Tuesday, we will be talking about the rest part of the homework and also the rest softwares that you might need for doing this kind of chromatin analysis. Okay, so if there's no other questions, I'll stop recording here.